All right. Well, you guys are getting settled. This is a good opportunity to go ahead and just pull up your Turning Point program from most likely your phone. The app is preferred, and join the session just so that when the first Turning Point slide comes up, you're already ready to participate. Saves time. Thanks.
Did you all guys get that?
They got it.
No.
Testing. Yeah, just make sure that you load your Turning Point app now and log in with your account and join the session just so that your devices are ready for when the first polling slide starts later on or a few slides in, so that you're not fumbling to get signed in and join the session. Session IDs on the presentation will remain at the bottom at all times. This is a good first go. Yeah. Did you register? No, from the app. You can, yeah. Or responseware.com.
So this is actually what's nice about this as a test, because in some classes as you go forward, especially in second year, you'll be using this for class quizzes and responses, okay? So it's best to kind of figure this out early on, so when you get down that road, there's no questions. Hopefully it works. We'll find out.
Yeah, it will, but some of them might be Wi-Fi.
Right. And I can bring up the poll now to see if it turns on. Should I do that? Now, the dental school. Okay. So we're going to begin. And what we're going to do is we're going to start with a little bit of review and continue with the epithelial tissue and go into membranes. As you see, everything is kind of a continuum. And if we had this cartoon up here, remember what we said is yesterday we talked about the apical side of cells, of the epithelial cells. We talked about the lateral side, those junctional complexes, so you feel comfortable with the order of the zonular occludence, or the tight junctions, the zonal adherence, and then the macular adherence, aka desmosomes. And we also introduced gap junctions. Some of these players you will find in other cell types, not just epithelial cells. For example, desmosomes, gap junctions, a form of zonular adherence. These will be found in other cell types. The zonular occludence, the tight junctions, you think of epithelial tissue. So sometimes it's just restricted. Other times it could be found in other places. We talked about a little bit about the basal side, where you have, if you have this as being your basement membrane. Dr. Newman will talk more about that with extracellular matrix. The basement membrane has a specialized extracellular matrix components. Basically, it contains the basal laminar, which is the material produced by the cell that sits on it. In this case, the epithelial cells producing the laminin, producing the proteoglycans, producing collagen type 4. And he'll go into that more. The connective tissue that it's linking to underneath is secreting other types of collagens, forming that reticular laminar. Together, that makes up the basement membrane. Cells like the epithelial cells have receptors like this hook that's attaching to this basement membrane. The ones that we talked about, the hooks basically are integrins. They're the adhesion molecules. Integrins have the property of having, okay. The integrins have the property as being a dimer of 2 subunits. And in this case, it's attaching, one end is attaching to the basement membrane, shown down here. And in the other case, we have it attaching to, inside the cell, intermediate filament proteins. As soon as you see intermediate filament proteins attaching to an adhesion complex, you think of a desmosome, or in this case, half a desmosome known as a hemidesmosome. It allows cells to withstand mechanical stresses. Okay? Makes sense? Okay. So that was a little bit of what we talked about when it comes to the basal surface. And what we did is we stopped. Okay, this was here, we were talking about it, is the desmosome and hemidesmosome. Here, you can identify it using electron microscopy because of the dense staining plaque that is identified by these arrows. So you can imagine that this is the plasma membrane, this is the hemidesmosome, and that there are little adhesion molecules that are attaching to this basal laminar. And here are the reticular fibers, and together this is the basement membrane. Okay? Okay. Now, the additional properties of epithelial tissue, just want to go through quickly, is epithelial tissue is regenerative. So they have mitotic capability. So you may ask, okay, what has nature done to protect those cells undergoing mitosis? Well, you can imagine when you have an epithelial lining, once again, we always talk about having a lumen. So here's your lumen. And you can imagine that outside in that lumen is fairly harsh environment. So what nature has done is protected those stem cells that are going to rejuvenate the epithelial lining when these cells out at the tips get damaged or sloughed off. And what nature has done, in some cases, you find these projections, the architecture forming these what's called villi. Okay, so these villi is a larger structure. Microvilli are apical projections on cells. Villi is where you have the projection of the epithelial tissue and the connective tissue core, increased surface area to even a greater extent. Okay, so what nature has done is that you have these villi, and these villi actually extend into the underlying connective tissue known as crypts. And nature has put the stem cells in these crypts as a protective nature. So when you think of from a cartoon perspective, okay, you would actually see a cartoon where you have stem cells in that crypt area. Here is a stratified squamous epithelium. Okay, the apical cells are squamous. It's stratified. which means do all cells sit on the basement membrane? And the answer is no. Okay. Down here, now you don't see the basement membrane, but there is one. We need special stains. And what nature has done is at the very base, probably here, would be mitotic cells. Okay? The chromosomes are highly condensed. So in this boxed area is a higher magnification, and you can see what was captured is a mitotic cell right at the base of this epithelium. Once again, away from that lumen, which can be quite harsh. And the idea is that the cells would replicate, and they get a migration of cells towards the apical end of that epithelial lining. Make sense? So in the module session, you'll be asked, where would you identify mitotic cells, right? And so in, say, a stratified squamous epithelium, it would be in this basal layer and so forth. Okay. And this just shows it in a cartoon perspective. You would have these mitotic cells, these stem cells, and they would be differentiating as you go towards the apical side of that membrane. So again, always think about that cartoon to help you image what's being talked about. Now, the other thing I want to quickly go into are glands. We talked a little bit about glands being epithelial tissue. There are two types. Well, there's two major classes, endocrine and endocrine glands and exocrine glands. Endocrine glands, which is a type of epithelial tissue, secretes its product into the vasculature. Okay? Exocrine glands will secrete its product into a duct to make its way out into the bigger cavity of that lumen. Make sense? So we talked a little bit about that, but I want to say that glands, the exocrine glands, also come in two flavors. One is unicellular glands. The goblet cell is the best example. So here is the epithelial lining. Here are your goblet cells. These will secrete its product right into the lumen. Those are unicellular glands. Okay? In contrast to multicellular glands, which we'll talk about in a second. Okay? Now, with secretion mechanisms, when we talk about exocytosis or the release, there's actually three mechanisms from a histological perspective that products can be released from cells. One is what's called merocrine. That's normal vesicular trafficking and exocytosis. Dr. Fried will talk about that further. Okay? But merocrine just means vesicles contain the product, fuses with the plasma membrane, and the material is released. In contrast, apocrine secretion is the product is actually formed in the cytoplasm, like lipid product. And that product then is is released into the lumen with a portion of the plasma membrane. So the membrane pinches off with that product. Okay? That's called apocrine. There's only one area in the body that has that type of secretory mechanism. What would you say that area is? I'll give you two answers. Either apocrine glands of the skin or the mammary gland. What would you say? Mammary gland. Can't fool you guys. Okay? The apocrine glands of the skin are a misnomer. Okay? Misnamed. Holocrine is the third type of secretion where the cell, it's a form possibly of apoptosis. The form just opens up, the cell just opens up and dumps its entire product out. And we'll talk about that when we talk about skin. Okay? So sebaceous glands. When we talk about types of secretion, we will talk about serous, and you'll see that even in the epithelial lecture lab, a module session. Okay? Serous means that you have a watery, non-glycosylated or poorly glycosylated proteins, so it picks up that stain quite readily. Versus what we talked about, mucus. Mucus is an extensively glycosylated protein, poorly stained by H&E, but visualized by PAS. And we showed that yesterday with the goblet cells, right? So you will go through, and here are your unicellular glands. Okay, these are containing mucus because it's picking up the H&E quite poorly. So use your stain to identify what you're looking at. Okay. Here, when we talk about multicellular glands, and we introduced that a little bit yesterday, We have secretory units. They dump the product into a ductal region, and that ductal portion is a conduit or can remodel the product. You'll hear about it, and will dump its product into a lumen. There could be multicellular, meaning there's multiple cells. You could have multiple acinar units. Each one of these secretory units is sometimes called an acinar unit. So you'll hear that term, and it will dump its product. Now you notice that this acinar unit has circular nuclei. This acinar unit has flattened nuclei. You see that? Well, that actually will tell you what's in that cell, what product is being released. It turns out that the cells that have circular nuclei are secreting a serous product, whereas with the flattened nuclei, the granules push the nuclei to the basal end, flattens it out, and they are typically mucus type of products. So in your module session, you're asked to look at glands and identify whether they're serous or mucus, and you can appreciate down here that these are very pale staining. Nuclei are pushed to the basal aspect. These are all mucus-secreting glands. They will be dumping a product into a ductal system, and this is the portion of the duct because of the cuboidal to columnar type of epithelium. Here you can imagine that in this case, you notice the nuclei are circular. These are serous, secretory portions of serous glands, and they will secrete into a ductal system. And here's part of that duct getting out to the epithelial lining. You see that? So basically, it dumps its product into a ductal system, which is a conduit to get it out to the lumen. Okay? So I just wanted to give you a heads up. You'll be taking this further when you get into the module sessions, and you'll be seeing secretory units and ductal portions of glands all throughout the module sessions. Okay. So we're going to move on and talk about membranes. The plasma membrane, okay, you've seen the video online? Yes? Okay? It basically is rehashing a lot of what you've already known. So we're going to bring some of that in, and we're going to let you remember the other part. What I want to do is talk about the topography of the membrane. Now, the membrane, as you know, is separating the external environment from the intracellular compartments, right? It's just like intracellular membranes are separating the cytosol from other areas of the cell for specializations of function. So the plasma membrane, okay, here is your objectives, which you can read, okay, at your leisure, okay? The question is do you see the plasma membrane by light microscopy? And what's the answer? No. And so this chart kind of gives you a flavor that you have light microscopy. The resolution is about 0.2 microns. Here, the electron microscopy is about 2 nanometers. So if you look at the list, you see cells. This is cells can be as small as red cells are 6 microns. Platelets are fragments of 2 microns. You will see them. Oocytes can be greater than 100 microns. But the point is you'll see cells, you'll see nuclei, you'll see cilia, and you can identify microvilli. Okay? Mitochondria, lysosomes, and peroxisomes are really at the cusp of light microscopy. You don't see that unless you have special stains, or if there's just an overabundance, you get a flavor of it. We'll continue talking about that later on. But the plasma membrane here is 5 to 10 nanometers. Okay, so you just don't see that unless you use electron microscopy. So it all comes down to resolution, to be able to distinguish 2 particles from each other. So here, we talked about this yesterday, okay? But just from it, every time you see an image, it could be in class, in module sessions, on an exam, there is information given to you. Just from this, I'm telling you that this is the left-hand side. This is an electron micrograph of this type of epithelium. Okay? What are the apical projections? Okay? And why do you say cilia? Because BB is basal bodies. Okay? And what we've said is that the cilium, basically you have it's elongated, and we would probably give you size as well. These are much larger than microvilli. It could be around 10 microns. There are microtubules inside. If we did a cross-section, it would be a 9+2 configuration. Okay? And each cilium, multiple cilium on top of a cell, each cilium is an outgrowth from a specialized microtubule organizing center called basal body. Okay? Now, if you look at the level of the light microscope, okay, are the basal bodies imaged here on the right-hand side? Yes or no? Yes. Okay? Here, the basal bodies are this line. Now, why are we getting a line here on the right-hand side? What's that? Resolution. It's not good enough to discern individual basal bodies. So when you go to the LM, what you get is a single line that goes across. You see that? Okay? So in some cases, we know that we have, here's the cilia, this is the basal bodies, this is the basement membrane down here. Okay? And the reason why it's a line, again, the resolution is too low to be able to discern individual basal bodies. So it gives a line across. Make sense? So that's when we talk about resolution. Okay. So when we look at cells and we look at plasma membranes, so there are clearly every portion of a cell is surrounded by a plasma membrane. Okay? Cilium, plasma membrane. Microvilli, it's a continuation of the plasma membrane, right? So there's always surrounded by plasma membrane. In some cases, we will see boundaries between cells. You are not looking at the plasma membrane. The plasma membrane would exist there of adjacent cells, but it's not the plasma membrane proper. Okay? We would have to go to the electron microscope. Okay? And here's an electron micrograph of a plasma membrane of a cell. And again, it's the trilaminar structure. Electron dense, electron lucent, electron dense inner portion. Okay? This is the outer portion of the cell, inner portion of the cell. So you would have to be able to image it at the level of the electron microscope. Now, we talked about in the pre-lecture video that you have your major players of phospholipids form that core. We talked about the lipids, major being the phospholipids of setting up the scaffolding, but they're also important for functionality. And you recognize that you have phosphatidylserine is negatively charged, phosphatidylnositol is negatively charged, and then you have a distribution of phosphatidylethanolamin and phosphatidylcholine. You should be able to look just at the distribution of phospholipids and the glycoproteins and GPI-linked proteins and all these players to know are you looking at the inside or outside of a cell. Okay? One thing is that all these molecules are amphipathic, means that they have both positive and negative charges. So if you throw this into an aqueous environment, it naturally will set up a lipid bilayer or to be able to shield a hydrophobic portion of the phospholipids from the aqueous environment. And that's all explained in that video. We talked about there that you have the phospholipids. The major phospholipids are the glycerol-based phospholipids, phosphoinositol, phosphatidylserine, choline, ethanolamine. And then you have the sphingomyelin, sphingolipids, which we'll talk a little bit about later. We'll talk about glycolipids, always facing outside the cell. We'll talk about that a little bit later in the course. Okay? Then we'll talk a little bit about peripheral and integral membrane proteins a little further, and the glycocalyx. And all these are discussed either to a greater extent on that video. So what is the orientation of membranes? And so when you look at this, you should be able to appreciate, you look at this, these two membranes, one on the left, one on the right, I would tell you that just looking at it, one of them does not exist in nature. And you should be able to tell me which one. Okay? But we're not going to do that right now. Okay? You can look at that and just convince yourself. But here is a certain type of scenario. A 40-year-old was admitted to the emergency room with complaints of shortness of breath and leg swelling. Ultimately diagnosed with left femoral vein thrombosis. So as soon as you think of thrombosis, you're thinking of activated platelets. Okay? And pulmonary embolism. So those activated platelets form the clot, move to the lung. The platelet ultimately acquired an enlarged spleen, and also you're getting a decrease in circulating platelets. Why? Because they're all becoming activated and forming a clot. Okay? Activities of protein C, protein S, and antithrombin were found to be normal. We'll talk more about that in the coagulation lecture in biochemistry. Antiphospholipid antibodies were detected, and following other tests, the patient was diagnosed with an antiphospholipid syndrome. You analyzed the topography of the plasma membrane of platelets and erythrocytes from this patient and observed increased annexin 5 binding using flow cytometry. Okay? Which of the following membrane is representative of this patient's cells? This is the polar. Okay? So you have A. Okay? As I get older, the print gets smaller. Here you have, what's that? Excellent, 2 responders. Isoprenylated protein shown here, GPI-linked protein, N-linked glycosylated protein. Okay, here, isoprenylated protein. Okay, N-linked glycosylated protein, GPI-linked protein. So you see, you have all these players. So now, you can talk to each other, by the way. This is not a test. You're not being graded on this. Okay?
This is the part where they, how do I join this?
Well, good. This is it. I love this when you get the sound.
So yeah, you make a good effort to make them force it to collaborate, which
Yeah.
is great.
Yeah. And hopefully the distribution's all over the place. You know what I need to do next year is put it on their laptops, the picture, so they can actually see it. But in a way, this is nice that they're all pointing up here.
They can see it from the device.
Oh, and the device. Oh, got it.
Whatever device they use, laptop.
Oh, okay. So I see. All right, so yeah, see, as soon as I say something, it speeds up. Went from 39 to 50.
I think they're really thinking hard about before making their selection.
I think so.
When they get into the board, when they get seasoned, they'll be able to answer that.
I always make sure I put the right number underneath.
Right number what?
You know, the way I do it, I hope I do it right, so the response correlates with the right number or letter.
Oh, I see. Well, I hope so.
I
No,
get-
yeah, you're right.
I know it's tough each year, so I get frantic.
Well, I mean, as long as you identify which one's the correct one.
Right.
in your presentation.
By the way, we're being taped.
Wait, how did you?
Okay, so the point here is a couple of points here. A, we're going to come back to this question also, but we'll see what the distribution is. But what's also very important is that we have, it keeps going as I turn around. Just want to make sure, as I mentioned, get used to using clickers. I think in Block 2 they're going to be using it a lot more. I don't know if they're going to be using it because you are registered, so some classes will use it as part of a testing tool. We're not doing that here, okay? So make sure that when we have clickers, and we'll have some more coming up, get comfortable with it so you work out the kinks now. So what we're going to look at is, okay, so we're going to switch. Last minute signal. Well, we have a pretty good look. So we'll see what the distribution is. Okay. So let's see if, so C, what you're saying is that this is in this group, right? That's what, if I did everything right with my numbering, we'll find out to see if it stays the same. If it stays the same, then we know that that's the answer. If it doesn't stay the same, then we know it may have shifted, okay? So now let's go further, because the key question is, what is annexin, right? I mean, that's an open question, okay? What is flow cytometry? So there is information that's given. You know, here what you're saying is with C, N-linked glycosylated protein, isoprenylated proteins exist on the same side of the membrane, right? I mean, that's really what's saying here. And GPI-linked is on the opposite side. It's also saying phosphatidylserine is on both sides of the membrane, phosphatidylnositol only primarily facing in, with the N-linked glycosylated portion, right? That's what's, if you break down what's being said. So let's now dissect out further. So, first of all, let's look at the lipids that are in membranes. So when we look at that, you notice that inside the cell is primarily free phosphatidylinositol and its metabolites, phosphatidylinositol phosphate, phosphatidylinositol bisphosphate. Why? Because phosphatidylinositol phosphates are extremely important in signal transduction. Okay? So you probably remember from studying for the MCAT and so forth, phosphatidylinositol metabolites are broken down, forming IP3, inositol trisphosphate, diacylglycerol, calcium mobilization, and PKC activation. We'll go into that further. So whenever you see phosphatidylinositol, it's always facing inside the cell. That's the free phosphatidylinositol and its metabolites. Phosphatidylethanolamine, phosphatidylcholine varies depending on cell types. You saw the distribution in the video. Phosphatidylserine is primarily on the inside of a quiescent resting cell. Okay? There are occasions, very important times, when phosphatidylserine is flipped out across the membrane. Okay? So that kind of sets up. So phosphatidylinositol is primarily on the inside. So you don't see it on both sides, the free phosphatidylinositol. So, and that's because, again, phosphatidylinositol metabolites, when you tickle receptors, you can activate certain enzymes like phospholipase C, forming a breakdown and generation of second messengers, which allows cells to respond to the external environment. And we will go on this further when we talk about signal transduction in the course. Second point, phospholipids, you have a lot of lateral diffusion, but you very rarely get flip-flopping. You don't get it spontaneously. You need help. So it's facilitated by specialized proteins. There are proteins called flipases, which kind of set up that asymmetric distribution of phospholipids across the membranes. And scramblases will actually bring phosphatidylserine from the inner leaflet to the outer leaflet of the plasma membrane under the appropriate conditions. So what do we say? So normally in a non-activated cell like a platelet, which is a fragment of a cell, you could immediately see that this is the inside. Why is it the inside? If I remove the term cytoplasm, because you have cytoskeletal structures, right? And so what this is saying is that phosphatidylserine, the negatively charges, is primarily lining that inner leaflet. Occasionally you see one out there. You may stimulate a cell like the platelet, and what happens? You have phosphatidylserine flips out because of the activation of certain proteins, right? Now, this is going to be very important. Well, in the activated platelet, that negative charge sets up a docking station for the clotting factors to sit down. Okay? So when I come back and talk about coagulation in the biochemistry course, we're going to ask you, what is the remodeling, what leads to an increased negative charge of an activated platelet? And you're all going to yell out, phosphatidylserine flips out. Okay? It's crucial. There's only, though, from a medical perspective, there's a disease called Scott syndrome where they believe the scramblase is inactive, and so you don't get this flipping out. It's a bleeding disorder. You will never see this because there's only one patient that has been diagnosed with it, to tell you the truth. So, but it's extremely important component of, say, platelet activation. Aging, red cell aging, one of the theories is that it's the flipping of the phosphatidylserine recognized macrophages. Apoptotic cell death, very important for normal development processes. And you know, when apoptosis, phosphatidylserine flips out and allows it to be cleared by macrophages. Okay, so that's one piece of the puzzle. So, part of the STEM was you analyzed the topography of the plasma membrane of platelets and erythrocytes from this patient and observed increased annexin 5 binding using flow cytometry. Which of the following represents the patient's cells, right? So when we look at that, we have to say, well, what is annexin 5? Well, first of all, annexin 5 is a membrane impermeable protein. And what they've done is that they basically have taken it and they made it fluorescent. They put a fluorescent tag on it. It's only going to recognize proteins that are expressed on the outer surface of the plasma membrane because it can't penetrate that plasma membrane. Okay? Or I should say it's only going to recognize entities that are on the outer surface. So it doesn't have to be a protein, it could be lipids. So here, what you can do is if you took this cell and you incubated it with a membrane-impermeable fluorescent probe, but it wants to interact with this entity, it can't because it's shielded by that plasma membrane, right? And if you take it and you put it into what's called a fluorescent activated cell sorter, which identifies cells that are fluorescently labeled which are not fluorescently labeled, you will find that your your whole population of cells are not being labeled. This is number of cells, and this is fluorescent intensity going in the x-axis. Okay? So we can extend this x-axis out like this, and so we have, in this case, only unstained cells. So far, so good? But if the cell gets activated in a way and you flip out that component that's recognized by the green fluorescent probe, now you can get an increased labeling of cells, right? So why do I tell you this? Because we can put names to this. Annexin actually recognizes phosphatidylserine. And so in the resting state, platelets and red cells, if you were to incubate them with annexin, fluorescently labeled annexin, you would not get any labeling. However, in this patient population, you have activated platelets, phosphatidylserine flips out, and annexin now can recognize those activated cells, and you get a fluorescently labeled population of cells. Phosphatidylserine, yes. So it's a tool that's used as to look at the labeling properties. Annexin 5, specific for phosphatidylserine. Okay? So that kind of explains that portion of the question. It means phosphatidylserine has an increased annexin binding, right? So when you look at this, okay, I'm not sure if it helps you much. It says increased annexin 5 binding. So let's do it again real quickly. So your eyes should be looking at phosphatidylserine distributed on both sides of the membrane, right? And so you would immediately either stay with your response or possibly switch it. But let's see. So we didn't give you any more information. We helped a little bit. Okay, so you can do this fairly quickly. Okay, so hopefully that has helped a little bit. Okay, and so let's see what the distribution is now. You notice I kind of talk a little slower to get those individuals caught up. I think there was 210 people before. Okay. Now we shifted to B. So it's interesting. 54% said B. 33 is staying with C. We still have a little bit with A and D. Let's continue with some of the story, right? Okay, this is how you're supposed to dissect out these questions. So let's go further. So let's talk about proteins, peripheral versus integral. You had it on a pre-lecture video. We talked about the peripheral proteins associated. We'll come back to that. Integral membrane proteins are basically spanning the membrane. In most cases, we're talking about the amino acid backbone either spanning or partially embedded. Could be multiple spanning and so forth. But when we looked at a typical, this is a cartoon of an integral membrane protein spanning one time. If I didn't have the word cytosol, which means cytoplasm, you would be able to look at this cartoon and tell me immediately what's outside the cell and what's inside the cell, right? Because you know that N-linked sugars are always facing outside the cell. Why is that? We'll talk about it when we talk about in the lecture. I'm running this whole lecture without lights. Hello. When you look at it, N-link glycosylation is initiated in the lumen of the endoplasmic reticulum. What's in the lumen of the endoplasmic reticulum gets into the lumen in secretory vesicles. Okay? It goes into the Golgi, and it's in the lumen of the Golgi. And the idea is that what's in the lumen of the Golgi, okay, buds out, becomes the lumen of secretory vesicles. And what's in the lumen of secretory vesicles, when that vesicle fuses with the plasma membrane, what's inside now is facing outside the cell, right? So N-link glycosylated proteins are always facing outside the cell. So that kind of sets up an architecture. Disulfide bridges are typically facing outside the cell because inside the cell you have a reducing environment, so you typically have free sulfhydryl groups. Okay? So this kind of gives you an idea of topography of the plasma membrane proteins. Okay? Now, I should also mention with membrane proteins, proteins are dynamic, by the way. Okay? So I'm going to take a regression from this little vignette. Proteins are dynamic, meaning that here is endothelial cells. So if it's endothelial cells, simple squamous epithelium lining the lumen of blood vessels. And they are non-thrombogenic. Platelets don't adhere to them. You have policing of leukocytes. Now, if you have a site of inflammation, so you have some type of bacterium or some damage underneath in that wall of that blood vessel, you can get factors that will stimulate the endothelial cells to now express specific surface receptors like selectins. And selectins now will attract leukocytes, okay, white blood cells, which will then evade the wall to go after that site of infection. This is basically saying that you have upregulation of proteins like selectins during inflammatory responses. And this is a theme that we will continue to come back on. So you can have expressions of proteins on membranes at different times depending on the state of that protein, of that cell. In other cases, you will have proteins that are expressed all the time but on different cell types. And so here is an understanding families of molecules present on different cells, provides tools to distinguish in diagnosis and therapy. So we're going to start talking about CD molecules. CD, cluster of differentiation, identified on leukocytes. Okay, now they can be on other cells. But when we start talking about like CD20, this is a marker of B cells. And we're going to talk about it a lot. CD19, another marker of B cells. And you have all these different family of CD markers on cells. This comes important, so when someone says, let's do a differential diagnosis and let's look at CD markers, you may get a flow cytometry report. You now know what flow cytometry is. This is of a patient that had a form of lymphoma. Okay? And here you have here is CD26, CD4 positive. You will know what CD4 positive means. Okay? Here's CD19, B cell markers. Here are some other surface expressed markers. Here's CD56, natural killer cells. And you can see this patient actually was very high in some of the B cell markers. So CD markers, we're going to start introducing various terms throughout the course. but really appreciate that these are surface proteins that are resident on specific cell types. Okay. Now, let's go back to the vignette. Other type of anchoring of proteins to the membrane includes proteins that are anchored via little fatty acyl groups. Palmitic acid is one of the examples. Myristic acid, farnesylation, isoperenylation. What happens in this case is that these proteins are all made on free ribosomes in the cytoplasm. Okay? And after they're produced, or while they're being produced, basically a little fatty acid tag is put on them. And so therefore, if they are to reside in the plasma membrane, will they reside in the inner leaflet or outer leaflet of that membrane? Has to be in the inner, because they are not going to be able to make it to the outside portion. So these guys will always be sitting inside the membrane. In contrast, a protein that is anchored via a glycerol phosphatidylnositol linkage, a GPI linkage, okay? I will tell you that that linkage is put on in the lumen of the endoplasmic reticulum. What else was modified initiated in the lumen in the endoplasmic reticulum? End-link glycosylation. Where did end-link portion of the glycoprotein exist? Inside or outside the cell? Outside the cell. So therefore, where do you think GPI-linked proteins always exist? What's that? Outside the cell, right? So whenever you see GPI-linked proteins, they're always facing outside. So you say, well, okay, why do I need to about GPI-linked proteins? Well, here is an example, PNH, okay? This is patients who will present having hemolysis with red blood cells as a consequence of complement activity in normal serum. So complement is a set of proteins that circulate in the blood as an inactive entities. And when you have typically bacterial infection, these complement proteins become activated, form pores in the bacterium, and leads to lysis. Okay? So you say, well, if they get activated, do they harm on our own cells? Well, if you look here, this is showing that for some reason, at night, that this is actually showing that here, if you take the patient's blood and you spin out the red cells, the serum should be yellow clearish. But these patients, the serum is red. Why? Because the hemoglobin leaked out of the red cells in these patients. And so what's happening is that this is a disease where you have sudden night hemoglobin in the urine. The urine gets into the hemoglobin gets into the urine. It could be life-threatening. It's acquired. Okay. So you have hemolytic anemia. You can also have platelet activation. And it's a genetic defect in a gene in the bone marrow. And that gene, the PIGA gene, is responsible for attaching the phosphatidylnositol linkage to proteins. Okay? And it turns out that on red cells, there are two proteins, CD55 and CD59, some of the CD markers. And it turns out that normally complement, if they get activated, form these channels within cells and can lead, if that happens on the red cells, it will lead to the leaking of hemoglobin out of the red cell. But nature has been very clever, and what they have done is they have placed CD55 and CD59 on the plasma membrane of red cells and of platelets. So if you get activation of complement system, what happens is now these cells won't be lysed. But in these patients, they have a defective machinery, so they can't put GPI linkage on proteins. So you won't have CD59 and 55 expressed on the outside. And so now what happens is complement, when it form channels, and you get hemoglobin coming out of the cell. Make sense? Okay? So GPI linkage, very important for placing proteins on the outer leaflet of the plasma membrane. Okay? And so you can actually do a flow cytometry with these patients to see, do they have, say, CD59 Okay, by you have fluorescently labeled antibodies. You take the patient's red cells, incubate with the antibodies, and ask, what does it look like? And you either have individuals that look like normal. So here is a flow cytometry. Here you have cell count. Here you're looking at fluorescence intensity. This type 1, normal intensity, a lot of fluorescence, so they have CD59. No fluorescence in the type 3 population, because this is almost no fluorescence. This is your patient population without the CD59 being expressed. They have PNH. And then you can get a gradation in between where some of them have a lower level being expressed. Okay, does that make sense? So now here, this is how you can use flow cytometry to be able to evaluate data from your patients. So based on this information, now let's finish this out. You have increased annexin binding, flow cytometry, look at the distribution of phosphatidylserine, look at the distribution of N-linked glycosylated proteins and GPI-linked glycosylated proteins. Are they supposed to be on the same side? And you would say, okay, so now what will your response be? Yes, question? So this is GPI-linked protein. This is N-linked glycosylated protein. This is a GPI-linked protein, like P50, like CD59, and this is an N-linked glycosylated protein. Here's your GPI-linked protein. Here's your N-linked glycosylated. Help you? Okay, so let's see what we got here. So we have a good response rate, and we'll see if the distribution now has changed. And we, okay. Excellent. One topic you don't have to study, right? But you see how you can break that down, okay? Just from a simple cartoon. Everything that you will see, there's some information that's yelling your name or telling you what the answer is, okay? So if you can learn any topic, just like we dissected this out, this is what module sessions are supposed to be. This is what lecture time is supposed to be to help you. So when you go out, now your day's not cluttered, okay? So this is the exact response that you would like to see, okay? Great. Thank you. So the last thing we just want to go through is, okay, let's talk about the glycocalyx. The glycocalyx, as we talked about in the pre-lecture video, is a sugar fuzzy coat. Now appreciate that you will see, here's an electron micrograph, a transmission electron micrograph we showed yesterday, epithelial cells, junctional complexes. Here's your microvilli, and this gray staining region is your glycocalyx, okay? And as you know from that video, it's the adsorbed glycoproteins, or it's the sugar groups on the integral membrane proteins. From a physiological perspective, it's extremely important because it's protective of that surface of cells. Like in the endothelial cells, it protects and it prevents cells from adhering, like platelets from adhering, because it's a very negatively charged surface, and platelets have a very negatively charged surface as well. There is a constant shedding and replenishing of the glycocalyx, And in some cases, under pathological conditions, you can get an excessive amount of shedding, and now you can lead to damage of that external membrane of cells. Okay? And so we'll go into that further when we, again, talk about inflammatory responses. So what you have is recognizing sugar moieties are glycoproteins known as lectins. Okay? These are sugar-loving proteins. And the reason why I show you this is that means that these lectins are receptors. Receptors for specific sugars. Now, why is that important? Because you can imagine that you have lymphocytes that have different specificities. You have lymphocytes that are going to be home to the GI tract. You may have lymphocytes that are being home to the respiratory tract. Well, how do they know? Well, the lymphocytes may have different types of lectins, okay, these sugar-loving receptors on the membranes, and they recognize different sugar moieties. So the blue lymphocytes are going to, say, peripheral lymph nodes, okay, say, of the respiratory tract, whereas the red lymphocytes may be home to regions in the digestive system known as the Peyer's patches. Okay? So there's recognition. So the sugars are active recognition sites. So functions of the sugars: recognition, repulsion. I just told you, like platelets are negatively charged, and the endothelial cell surface apical side is negatively charged. So it's repulsion. Adsorption. In the digestive tube, you have entities that are adsorbing to allow for digestion. And protection, protecting proteases from cleaving proteins on the membrane surface. Okay. Last things I want to say is, membranes are fluid structures. So if I fuse different cells together, mouse with human, over time, these proteins will kind of go around, right? So they're very fluid. These are blood cells. So these are cells that are in circulation. But remember, there are ways to restrict the movement of proteins and lipids on surfaces. Here we talked about molecular fences. We talked about the apical junctional complexes, restricting movement of proteins from the apical side to the basal-basal-lateral surface of epithelial cells. But also appreciate that all these systems that we talk about don't live in isolation. Plasma membranes don't live in isolation. They're communicating inside the cell. We talked a little bit about adhesion, right? The integrins. So here's an integrin, right? Two subunits, alpha and beta subunit. Here, the integrin is binding to extracellular matrix proteins. Dr. Newman will talk about this further. And in some cases, we talked about with the hemidesmosomes, it's interacting with intermediate filaments. In some cases, you may have integrins interacting with actin cables. And the idea is that if you pull outside the cell from these changes in the extracellular matrix, it actually will pull on the integrin, which may pull on the actin cables. So you can influence cytosolic components via these complexes. Okay? Okay. Specialized regions of plasma membrane. We talked about apical and basal. We talked about tight junctions, adherence junctions, gap junctions. Dr. Ellinger will talk more about coated pits. Dr. Fried, in the bladder, these are the vesicles that allow for distension. And you will hear more later on about lipid rafts and caveolae. These are structures that are lipid, sphingomyelin rich, and important for signal transduction. So we'll bring that in later on. And the last thing is, with nutrients, plasma membranes are selective permeable barriers. And in many cases, you can have gases that can freely permeate, but you may need channels to get water in and out. Glucose needs channels, and ions needs channels and transport systems. And so the key is that we'll be talking about phagocytic properties throughout the course. You bring in large entities. Dr. Ellinger will talk about receptor-mediated endocytosis. So there are ways of getting things through that plasma membranes. But note that just by looking at the distribution of ions, sodium is high outside the cell, low inside the cell. Potassium is high inside the cell, low outside the cell, and so forth. So you should be able to interpret, are you looking inside the cell or outside the cell? We would give you normal concentrations. And you should need to know what's inside and what's outside. If I say actin, you would say inside or outside? Inside. If I say keratin, you would say inside? 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 Inside. Okay? So you need to be comfortable. What's outside, that's a responsibility of yours. Okay? The way molecules can get in and out is basically you have transport molecules. We'll talk more about them. And channels and carrier molecules. The key is that all these different transporters, which you hear a lot more in physiology, are going to be distributed appropriately on cells. Here's your epithelial cell. You saw this yesterday. Here's your microvilli, right? So you have certain transporters up here, and you have certain transporters down here. So the idea is that you have a transporter that you had a high sodium breakfast, excellent for when you're young, okay? You want to bring glucose, so sodium comes down its concentration gradient, high outside, low inside. It sucks glucose into the cell. Now glucose is high inside the cell, but low in the blood, so glucose can freely permeate through a specific carrier into the vasculature. You see? So that's where you have to know that polarity of cells are extremely important, especially in epithelial, be able to set up this type of gradient. Okay? So that's it. Now we link an epithelial tissue, membranes together. Great job. 5-minute break, Dr. Newman. Okay, and then we'll go on to the outside, the extracellular matrix and so forth. Excellent. Thanks, guys.
It was like Mission Impossible.
What was that?
It was like the movie Mission Impossible. It was just one thing after another.
There's a